about neurons mostly from the perspective of their membrane potential. But that's really only a tiny part of what's going on in the brain, right? And this is just sort of my story. So I'm faculty in the School of Public Health, but I care about neuroscience because, you know, there's a lot of things going on, neurological issues that are associated with aging and otherwise are going to become increasingly important to understand, right? They're not just network phenomenon. You know, we can get some insight into how the system responds to medicine by looking at upregulated channel activity and that sort of thing. But there's something on a different scale, different spatial and temporal scales that leads to axonal blebbing, the loss of myelin, dendritic pruning, formation of amyloid tangles, spreading depolarization and stroke, and that sort of thing. And these dynamics happen both inside and outside of the cell. And so for this morning session, I want to look into their story a bit and see, you know, and talk about how that relates to what we talked about earlier. So for example, here's a cell. This is from a model by Michele Maiori's lab. And it's available in ModelDB. And so when we talk about action potentials, right, we know sodium currents raise the membrane potential, potassium current lowers it. But what's a current? What does it mean when I say that there's a sodium current? I mean, as a basic physics question, what does it mean? Humanly.
There is movement of ions.
There's movement of ions. And the thing about movement of ions is that means you end up with a different number of ions on the inside and on the outside. So here's a question for you. How did Hodgkin-Huxley deal with that?
It doesn't.
It doesn't. Why is it okay that it doesn't?
Because the charge is so small that the balance is still there. I mean, the concentration inside and outside is still the same, or more or less.
So now, I mean, so that's certainly true. You know, you could imagine like we've got this 500 micron squid giant axon, right? It's big. But what if instead we collapse down to what we did last night, which instead of 500 microns in diameter, it's 1 micron in diameter. So that's 1 over 500 squared, the size. So now all of a sudden, 500 squared, 25 followed by 4 zeros, 250,000th of the volume, let's suppose, or you know. So it's a little bit more sensitive to changes if we're talking about a mammalian cell than a squid cell. And again, most of the time, so in that case, you absolutely do get concentrations that over repeated bouts would change things, except most of the time we don't care. Why do we still not care? Why can we ignore that most of the time?
Because there are things that aren't modeled, such as pumps that get the concentrations at the balance. Yeah.
Absolutely.
But if not, you have to model, no?
Yeah, exactly. So, and that's part of what we're going to talk about today. So here, for example, you know, we see this sodium current means sodium ions are rushing into the cell. Potassium current means potassium ions are rushing out of the cell. If you're, you know, trained in math, you can be mildly annoyed that you get more stuff when the current's negative and less stuff when the current's positive. But we can just cope with that. One other thing that I'll point out, fun fact about this cell. So here's the currents from this cell, and you'll notice that the sodium current peaks before and starts collapsing well before the potassium current does. Is that true in Hodgkin-Huxley? How does it look in Hodgkin-Huxley?
It's delayed. No?
This is, well, so in Hodgkin-Huxley, they're much more aligned. The sodium current comes on, and then the potassium current starts coming on later. It's coming on shortly after. But in mammals, there's a bit of a shift. There's a bit of a phase shift. Why is it advantageous that there's a bit of a phase shift in mammals?
And probably the activation curve is shifted, no? Maybe one reason.
Well, what's the downside of having sodium trying to pull you up while potassium is simultaneously trying to pull you down?
That there is a balance and you don't get the peak.
Yeah. If you want to get up to 40 millivolts or whatever, you're going to have to bring in a whole lot more sodium because meanwhile the potassium is constantly trying to pull you down. But if you phase shift them, sodium's not fighting the potassium. So you can move less ions. And if you move less ions, why is that a good thing?
It's energetic cost, no?
Yeah, so if you're moving less ions, right, you don't have, that means that the homeostatic mechanisms that maintain the cell's balance don't have to do as much work, right? It's easier to keep sodium, there's less sodium ions that need to go back outside, and there's less potassium ions that you need to bring back inside. And this matters especially if you're talking about thinner dendrites rather than, thinner axons rather than these large axons where, concentration changes can actually build up. So that was mostly a fun story, but yeah. So anyway, so anytime we have current, that means that ions are moving, and then so we might want to consider what's happening with these ions. By the way, so we've got all these homeostatic mechanisms. What's an example of something that would disrupt a homeostatic process and that would actually allow sodium and potassium to build up?
The sodium-potassium pump.
Yeah. So if something blocks the sodium-potassium exchanger, so like that, well, that would stop if like you ran out of energy. So like ATP, right? ATP needs oxygen. So a stroke, for example, could block ATP production, which could ultimately disrupt your sodium-potassium exchangers, which could be a problem. All right. So how do we actually deal with this? How do we find the relationship between current and intracellular concentration? And it's actually a pretty simple formula. It involves Faraday's constant. This is just a physics thing. So in 1 millisecond with a constant current, sodium changes by 10,000 INA times whatever the surface area is, divided by the product of Faraday's constant and the volume. Obviously, if you have a bigger volume to diffuse into, then your concentration change is not going to be as much. Again, Hodgkin-Huxley, big squid, minimal concentration change. And so, like I said, whether this has a big or a small impact is going to depend in part on the original concentration of the volume. And so in a squid giant axon, for example, your typical sodium concentration inside is going to be about 10 millimolar. Typical potassium is going to be about 54.5 millimolar. Anybody know if I have like a mammalian CA1 pyramidal cell or something? What about calcium? What's calcium like? We got 10 millimolar for sodium, 54 millimolar for potassium. Anybody know about calcium?
It's got twice the charge.
Well, yeah, but what's the concentration? In a cell, if I pick a cell, you there, Mr. Cell. Wait, where's my cell? All right, I have a cell, right? And I just say, what's the calcium concentration in here? Is it more or less than sodium?
Less than sodium. It's smaller than the others.
By a lot, in fact. So typical calcium concentration might be something on the order of 100 nanomolar. So not millimolar, not micromolar, nanomolar. So much, much, much smaller calcium concentration. Yeah, so if there was a question about spines on the Slack earlier. If I was looking at a spine and I have a spine head that's, let's say, half of a cubic micron, and I have, in that spine head, I have a 100 nanomolar calcium concentration and half of a cubic micron. Who's the physicist, chemist here that can tell us how many ions that is? Avogadro's number comes in there somewhere, right? That's a big number. It's got a 10 to the 23rd in there. That's going to help, right? Anybody know? It's about 30. About 30 ions at that concentration inside of a spine head of half of a cubic micron. So we're talking about really small numbers when we're talking about calcium. And some of our logic is going to be affected by that if we really want to get into that. Just throwing that out there. We're mostly going to ignore that for the purposes of today, though. But anyways, so my point is, we have currents, we can use math, we can calculate changes in concentration. That's what I meant by this, how are things different if it's calcium rather than sodium? The math, oh, well, there's a factor of 2 because of the charge of calcium, but other than that, calcium is just really, really small. And what this means ultimately is that, and a neuron is not a transistor. In the sense that in a transistor, right, currents go in, currents go out. I expect all of the, I don't know, billions of transistors that are in your computer to do the same thing today and tomorrow. But a neuron is not that kind of a device, right? It's got ions in it. It's got stuff in it. A neuron has state. And every time that we talk about this, right, there's a lot that's going on in this neuron. It's got all these compartments that are sort of restricting diffusion. It's got concentrations that are different in the different parts. So that means that a neuron, one moment to the next, we talked a bit about synaptic plasticity a bit in my exercise session, mostly just to acknowledge that it exists. But, you know, a neuron's got state, and there's a lot that's going on in there. And that means that how they respond is going to be dependent upon their state. It's going to be sort of dependent upon the state of things in the brain. So here's an example. So let's talk about the suprachiasmatic nucleus. All right. So suprachiasmatic nucleus is about 50,000 neurons in human. So it occupies a volume of about 0.3 cubic millimeters. This is in the hypothalamus, directly above the optic septum. And so the details vary by species, and the hamster looks fundamentally different than the mouse and the human for some reason. But the suprachiasmatic nucleus is what is your circadian rhythm. It's the central clock, right? And so what that means is that you've got a firing rate rhythmicity of about 24 hours, and that rhythm is preserved even in the absence of external cues. The individual cells are somewhat less stable than that, but collectively as a network phenomenon, all the more reason we need to study networks. Stay tuned for the rest of the course. But collectively as a network phenomenon, you end up with this about 24-hour rhythm. And what's driving that? Why are we able to do this even without considering the input? Well, because we've got protein oscillations that are happening. Proteins that are being transcribed, they're migrating around, they're binding with other proteins, they're coming back and inhibiting their own translation. Whatever. Right, so you've got these protein oscillations. There's this intrinsic state that's affecting how they fire, even if they get the same input, or So, so that's just suprachiasmatic nucleus. You see phenomenon in the prefrontal cortex that involves sort of hyperpolarization activated graded persistent activity. So that's one of the things where basically they hyperpolarize the cell. So they have a pyramidal cell from the PFC. They hyperpolarize it. It starts firing faster. Okay, and I'm not talking about post-inhibitory rebound. I'm talking about something that's sustained for like 10 seconds. And then they hyperpolarize it some more, and then it starts firing faster. And then they hyperpolarize it some more, and then it starts firing even faster. It's a fun little set of experiments. And what they end up concluding is that this is, they think that this is linked to the H channels and some details in the state of the H channels tied in with intracellular calcium, which I know somebody brought up the other day as well. So there's all this state that's existing, and I just want to say, and it's also just the brain state as well, because you could think, for example, in following ischemic stroke, again, you have less oxygen in the extracellular space. You might also end up with more elevated potassium in the extracellular space. And if you're interested in that, there are people here, some of the tutors, Craig, amongst others, that can tell you more about that. So there's a lot of state that's going on that says that's going to modulate how a cell responds or not. So does that kind of make sense in general? Okay, so neurons have state. Cool, and it matters. So how do we actually model this? All right, so fundamentally, we use what's known as a reaction-diffusion system. And it's just a fancy way, mathy sounding way of describing what it is that ions and proteins and all of that jazz do. They either bounce around. Bill talked about some of this with stochastic stuff earlier, right? They either diffuse. Diffusion is just bumping into each other because of temperature, right? Molecules moving around, bouncing into each other, and then eventually there's enough movement. Or they react, right? Here's a molecule, here's another molecule. They can either just move past each other or bind. And if they bind, they might do something. And those are the two main things that molecules can do. They either interact, they either react, or they diffuse. By the way, if you're thinking about like mechanisms like dynin and kinin, all of these transport mechanisms, is that an exception? Is that a third thing that molecules are doing, or what's the deal with that? Well, I would argue that fundamentally it's still reaction and diffusion. It's just that the reactions are such that they establish an energy gradient that makes it easier for the molecular motor to move from one point to the other. But its actual movement is because it's being bombarded. It's in this soup of the intracellular space where it's being bombarded. And so it's a diffusion driven that's biased. All right. Anyways, so molecules can react or diffuse. Well, what does it mean if I say the molecule reacts? So imagine that I have something, this isn't the best example, but imagine that I have sodium and chloride. And so if I have these two ions, there's some chance that they're going to bump into each other and then bind, let's say. If I have twice as many of one of the types of ions, how does that affect my chances that one of these guys is going to meet one of these guys? Well, there's twice. Yeah, there's twice as many of these guys, then I have twice the chance of meeting. That's known as mass action kinetics. Mass action kinetics is the basic assumption that people make. The more of a thing that you have, the more likely you are to bind. Cool. And so it's proportional. So the change in concentration of, this is our differential equation. The change in concentration of sodium in this case is going to be proportional to the amount of sodium I have and the amount of chloride I have. Likewise, chloride is going to change in the same way, and then in this case, sodium chloride is going to increase by the opposite amount. The negative sign indicates that I get less. I get less sodium when it binds and becomes sodium chloride. By the way, you'll notice one fundamental relationship here. What's that? How is the total amount of sodium and the total amount of sodium chloride related? In this model, if there's nothing else going on.
They're inverse?
Inverse means something specific. So let me not technically agree with that as a mathematician. But they as one goes down, the other goes up by exactly the same amount. So the sum, the total amount that I have is constant, which is I think what you meant. Right? This, of course, you know as the law of conservation of mass. You start out with sodium. If this is the only thing that it can do, it's either going to be in the sodium or it's going to be in the salt. And that's it, right? So that's the law of conservation of mass. Obviously, this is not necessarily limited to things where you have an equal amount of stuff. So you could imagine like calcium chloride. Calcium, as was mentioned, has a charge of 2, which means if I want to end up with, I mean, so this is a salt, so it's an ionic reaction. But if that means that if I'm going to need to end up with a thing that's a 0 net charge, so since chloride has a charge of -1, I'm going to need 2 chlorides to bind with that 1 calcium. And then then I need it's going to be, the rate of binding is going to be proportional to the amount of calcium I have times the chance of me meeting the first chloride, which is Cl. But again, I'm going to square it because it's going to, I need 2 of them, not just 1. Does that make sense? Why it's a square? All right, excellent. The other thing that I'm going to say about physics, if something can come together, something can go apart. And all we have is that there's a very small, it can be easier to go in one way or the other, but technically speaking, every reaction is reversible, right? It's just that it may be of different degrees. So it's a lie to say that it's a forward reaction. Really, it's maybe the backward reaction is just small. Obviously, then if calcium is changing at this rate, then chloride is going to change at double that rate because for every calcium ion that's involved, there's 2 chloride ions that's involved. Does that make sense? All right, cool. Okay, and that's mass action. And that's sort of the fundamentals of how an individual ion or an individual protein might meet with another one, if all of the reactions are what's known as elementary. What do I mean by that? What's an elementary reaction as opposed to not that? So you might imagine, for example, that there's actually a sequence of events that happen. You might imagine that a protein is going to bind to some enzyme. That enzyme is going to cause it to shift conformation in some way. And maybe then once it's in that conformation, it'll shift conformation in another way. Like maybe it's not a single step. All right. Maybe some of these reactions are more complicated. And so then this brings us to, so fundamentally physics, this is what we have, but what we often have is this enzyme and a substrate maybe bind together, and then there's a reaction, and then the enzyme disassociates, and you're left with a new product. All right, so it's not that one thing suddenly becomes another. It becomes that way with the help of an enzyme, like a cat. And if that cat comes along and does its thing, the rate at which that happens is going to be proportional to the number of cats you have. Does that make sense? If I run, well, let me say this. If I run out of cats, if I run out of enzyme, if all of my enzyme is busy, and I need the enzyme to go from the substrate to the product, guess what? I'm not going to get any more, right? So the enzyme becomes a rate limiting thing in a lot of biological reactions. And so we don't end up with things that increase arbitrarily, but maybe we see the rate of product, increase is going to start leveling off because I run out, all of my enzyme is busy. Okay, and there's, again, I'm technically speaking, this is always reversible, but we're making the assumption that the rate is just so slow that it's not worth mentioning. All right, but it turns out that if I have these kinetics and I make certain assumptions, all right, and so basically this means that the amount of enzyme that's binding to the substrate is either not changing, it's not changing, in any meaningful way, then the rate of enzymatic reaction reduces down to this form. And you can see that as the amount of substrate that I have goes to infinity, I end up with the Km is a constant. So as this gets large, this sort of dwarfs, the bottom just becomes ever so slightly bigger than the concentration of substrate. And so what that means is this whole thing reduces down to Vmax, right? As S goes to infinity. A little bit of calculus there, right? Taking the limit. And if it goes, sorry, I need to ask you to leave now. Thank you. And as the amount of substrate goes to infinity, it starts leveling off at Vmax. Km is what's known as the Michaelis constant. If you look at it, you'll see pretty quickly that if the concentration is equal to Km, then you have 2 times the concentration on the bottom, 1 of the concentrations on the top, you end up at half maximum. So the Michaelis constant is the half maximal rate, or is the concentration at which the rate is half maximal. So enzymatic reactions can be modeled using Michaelis-Menten. There are certain assumptions in order for this to be true. People routinely ignore them. That's probably at their peril, but yeah. But in any case, so the enzymatic reactions can be reduced down to Michaelis-Menten. And like I said, this is different from a mass action because Michaelis-Menten is going to level off, whereas mass action will increase forever with a concentration. The other thing that you have in biology is cooperative binding. You could imagine that I have, let's say that I have a, calcium buffer. These are things that exist. I have multiple binding slots on my calcium buffer, my protein, for calcium to bind. Cool. Suppose for a moment, and some of them, and the actual physics of the calcium buffer is going to depend on whatever your calcium agent is. By the way, fun fact, if you're injecting calcium indicator dye, that's a thing that calcium binds to. It's a buffer. It's going to change the kinetics because you can't observe without changing things. So, you know, fun. But in any case, so once I've bound to that first slot, does it make it easier or harder for the second molecule to come in and bind? Because now the protein, you know, I mean, clearly it's not the same as it was before, right? There's a difference between a protein that has something bound and something that doesn't. So you would think it's either it's going to change it a little bit, at least. There's going to be some consequence of it. If it's known as cooperative binding, that means that the more that I have, you know, once the first guy's bound, it becomes easier for it to bind. So that's the n bigger than 1 positive cooperativity case. And you end up with curves where the substrate is very low, not much happens because your odds of having 2 things that are trying to bind are very small. But once you have enough of it going on, once you have enough of a substrate, it's going to start increasing rapidly until, again, it levels off due to the enzymatic stuff. So you get these sigmoidal shapes. And so positive cooperativity is an n bigger than 1. Negative cooperativity is an n less than 1, which is where it's harder becomes harder for things to bond. So you get all these different possible shapes of relationships between the amount of substrate that you have and the amount of production based on the physics of the individual cell, or individual molecule that's involved. And you can fit these. I mean, so there is an interpretation that links to the number of binding sites. It's somewhat it's somewhat generally false, but there is an interpretation that you can give on the biology, but I'd rather not go into that because of the assumptions. But in here, by the way, is what's known as the Hill coefficient. And again, Ka, you'll notice if Ka and S are the same, then again, we get this half maximal rate. All right, so if the concentration is equal to Ka, we're at the half maximal point. Does this make sense? Cooperativity, sometimes it's easier, sometimes it's harder, and this is what it looks like. And you can fit it. It's probably the most honest answer. All right, so that's reactions. In my group, at least, I know we did, and all of my answers are in the Google Drive from last night's homework. But so space matters. Neurons are not points. Here's an example of neurons. These are from Cajal, who was a famous, one of the first to actually draw books, just draw images of what neurons look like. And so yeah, so these are very much not points. I hope we all agree that these are not points. And what that means is that their concentrations can be different at different points. And as I mentioned, within the cell, the concentration, there are different spatial zones within the cell. The endoplasmic reticulum is basically there, the entire cell. The Berridge, I think, referred to it as the cell, the neuron within the neuron, because it's there, the entire thing. And you can actually get some regenerative activity on the calcium dynamics as well. So it's a lot of implications. But so then, how does the space matter, right? And space matters because molecules can diffuse. They can be transport, but as we said, transport is really just driven by diffusion and reactions that bias things in one direction. Okay. Well, how does diffusion work? Well, Adolf Fick in 1855 provided what's now known as Fick's law. And Fick's law says, roughly, if I have things that are the same, if I have the same amount of concentration over here as I do over here, well, on net, I'm going to have as much stuff moving from one side to the other as I do going the other way, right? So if there's no gradient, if there's no difference, then I don't get any net diffusion. But the bigger the difference is, the more of a net diffusion I'm going to get. And the amount of diffusion that I get is going to be proportional to the difference. The rate at which molecules move on net is going to be proportional to the gradient. That makes sense? I think it's, and yeah, so that got him a law named after him. And so then the second law says, so that's just the rate of movement. And then the second law says, well, if I know the rate of movement, I can actually make a statement about change in concentration over time. And where have we seen that equation before? Fick's second law. We've seen that equation, but with a different thing and in a different context, where the variables meant something different.
Cable equation.
That's the cable equation. Electricity, charge, travels in the same way, mathematically speaking, as chemical diffusion. And this is great because that means that all of our theory that Wilfred Rall and people like him developed in the context of how electrical propagation works also applies to travel in one dimension at least within a cell of a chemical wave. That's kind of cool, and this is another reason the ER and calcium functioning as a neuron within a neuron, because you get this regenerative calcium phenomenon that's very reminiscent of an action potential. All right. So, and diffusion happens anywhere by definition, right? Like there's diffusion in the cytosol because diffusion is just a thing that molecules do. It's as if there's more than one molecule and the temperature is above absolute zero. Spoilers, you can't get to absolute zero. So if there's more than one molecule, it's going to bounce into it eventually, and then it's going to move. It's going to diffuse. That's it. So it happens everywhere. It happens in the cytosol. It happens in the organelles. It's going to happen in the extracellular space. I just want to point out, of course, when I'm talking about the cytosol, remember there's stuff inside the dendrite. It's not just, you know, a tube of fluid. There's ER in there. So it's not the full cross section. It's just, well, whatever the free part is. And when you're talking about extracellular space, it gets really sort of important to consider this because there's a ton of stuff. If I take a chunk of brain, right, there's a ton of stuff in there. There's neurons, obviously, astrocytes. There's also blood vessels. There's stuff in there. And each, you know, these are all things that are kind of tentacly entangled, and it's quite a bit of a mess. All right. And so you end up sort of wanting to think about things like tortuosity. So tortuosity is, if I want to go from this point to this point, how hard is it for me to actually go that distance? Because I can't draw a straight line because I'm going to run into blood vessels and neurons and all of that jazz. So it's the change in the path length. So as a rough rule of thumb, figure it takes about 1.6 times as long to go from point A to point B as it were in the brain. And there are papers that you can look this up, tortuosity estimates. And obviously, it depends on various conditions, and it's not necessarily uniform, you know, but in every direction. But biology has a ton of caveats. But if you want a number, 1.6 is a good number. Volume fraction, a free volume fraction might be on the order of like 20% is the space which fluid where molecules are free to diffuse around, and all of the rest is filled with something solid that you can't diffuse through. So these are considerations that really, really become important in the extracellular space. And like I said, you know, molecules in the extracellular space are a way in which neurons are able to interact with each other and affect each other. All right. One other bit before I get back into neuron itself. So let's say that I have a certain distance. I have a molecule that diffuses at 1 square micron per millisecond. This is on the order of the kinds of things that we care about in neuroscience. So let's say I have a molecule on this that diffuses at this rate, and I want to move a distance of 100 microns. How long would I expect a molecule that starts here to wait until it gets over here? Well, if I'm lucky, it's being bumped into by another molecule, moves a little closer. Next one moves a little closer, moves a little closer. It could go straight there. It could just make a beeline there. But in all actuality, every time that it gets over this way a little bit, it gets pushed back. And so the further I have to go, I have just as much chance of being pushed all the way back. So the time is not proportional to the distance. Time is proportional to the square of the distance, because the further out I get, the more chances I have of being pushed back. And there's math, and you can prove all of this, but that's sort of the intuition. And so what that means is if I have a molecule that's moving at this perfectly plausible speed, for a distance of 100 microns, and 100 microns is a perfectly reasonable apical dendrite length, let's say. Those of you in my session, we talked about we did an experiment yesterday with an axon that was 1 centimeter long, and we commented about how long that was because we wanted to see the propagation. This is 100 times smaller than that. Yeah, 100 times smaller than that. So still, when you do the math, this is a thing that means that it's going to take 5,000 milliseconds or 5 seconds for that molecule to diffuse that distance. Okay, 5 seconds. That's not so bad. It's a while. I mean, you can do a lot of action potentials in 5 seconds, but it's not crazy. Okay, so we go up to that 1 centimeter long thing that we did last night. Well, that's 100 times bigger, but it's proportional to the square of the distance. So it's actually 10,000 times longer. So now we went from 5 seconds to 50,000 seconds. And now there was a song a few years back that taught us that there's 86,400 seconds in a day. So that's basically, what, 13 hours, 14 hours? Okay, and that's the distance of 1 centimeter. Okay, what's the longest axon in the body. Well, you got one that sort of connects your toe to your spinal cord, right? So that's not a centimeter, that's like a meter. Not if you're my height, but some people. So then that's now another factor of 100. So what's that? We're about like 50, 60 days. You can do a lot of action potentials in 60 days. So the punchline is pure diffusion by itself is really great for really small spaces, but it doesn't work so well for long distances. Here's another thing though. But remember that I said that the equations for diffusion are the same as the equations for membrane potential, the cable equation. So how is it then, if diffusion goes so poorly with distance, that action potential propagation, though, is the amount of time that it takes for an action potential travel, is proportional to distance? But it's using the same equations as something, well, the electrical spread is the same as this thing that is proportional to the square of distance. How does action potential cheat? How does it get around this problem?
It is active. Also, fibers are myelinated.
We'll say
Well, myelin's just going to speed up the constant. It's not going to. But yeah, but it's active.
The Y is not the axon, though, right? So the fluid, the concentration across ions, movement of ions across ion channels is what's providing the current, and then it's the voltage that opens the next one.
Exactly. And you can think of this like an ocean wave is the other way that I like to look at it, right? The water molecule does not travel at the speed of the wave. Water molecule mostly just goes up and down. The local membrane potential mostly goes up and down. Charge is mostly going in and out of the cell. But a tiny bit goes a little further, and that is enough to trigger the wave at the next spot. And that tiny bit makes it a little bit further, because there's always some amount that's going to go faster than this. And so it's this regenerative phenomenon, and you can do the same thing with chemicals as well. And so calcium waves inside of dendrites and pyramidal cells are exactly the same story. And again, that ties back into this whole Berridge's neuron within a neuron concept, but now with chemicals and a different spatial scale. All right, so that's really all I want to say about the backstory of this. So let's talk on how we actually do this, how we actually deal with chemical dynamics within neuron. And then after lunch and or whatever, dinner, after break, we'll come back and we'll do some exercises on this. So how do we actually do this in neuron? Most of our stuff, the actual objects that we're going to create, we're going to start out using a different import than what we did last night. From neuron import H, we're still absolutely going to need this. We're always going to need from neuron import H. If I'm doing reaction diffusion, we're going to do from neuron import RxD as well. Okay. Cool. All right, and if you're on Read the Docs, there's a section under user documentation called RxD tutorials. I believe every single one of these is actually a link to Google Colab. No, that's a lie. Every single one of these is a copy of a notebook and has a link to Google Colab. I think that's the thing. So in particular, you click on one of these, you say, oh, well, that's cool. Let me actually play with this. You can then go and hit the link to Colab and then run it locally yourself. All right. So there's a number of examples. We actually have a few more on the Neuron website itself that I've been meaning to transfer over. But if anybody wants a good project, it still hasn't done anything, and wants to do software development, let me know. But in any case, so what's in the RxD module, and why do we care about this? Well, the important thing for us is that it gives us the ability to declare new regions, right? So I could have a region that is the cytosol, rxd.region, soma.whole-tree. So that's everything that's attached to the soma. I can map this to inner end region I, which is a little bit funny, but you remember that yesterday we talked about NAI versus NAO. So this region that's just inside the plasma membrane versus just outside the plasma membrane. So here in this case, I'm saying that the cytosol, the concentration of the cytosol is the concentration of sodium, for example, that sodium channels should see. This is as opposed to the concentration in the ER. When I'm defining the ER, it doesn't map to a traditional region. Because how do I want to say this? If you're inside the ER, then you're not inside the cytosol, which means you're not coming into contact with a channel in the plasma membrane, because there's the ER membrane. There's an ER membrane that keeps you from bumping into the plasma membrane, right? So, so the concentration inside the ER doesn't directly affect the ion channel activity, except through movement through channels on the ER. Same story for mitochondria, obviously, or really any other organelle, but you know. So this allows us to use declare arbitrary domains for whatever regions of interest that we care about. We can create a species, and then so in one line here, I can say calcium is present on the cytosol. It has a certain diffusion constant. This is in square microns per millisecond. If I was a better person, I would have included the units. And it has a charge, right? And so this gives us, so in 2 lines of code, what we've done is we've declared a domain. We've declared a molecule within that domain. We've allowed it to diffuse. And so in particular, once it's diffusing, and since we know the charge, and since we know that it's in the region that the ion channels are interacting with, that means that any current that comes in through the ion channels is going to change that guy's concentration. So now all of a sudden you could imagine that if instead of calcium here I had sodium, and I just did exactly the same thing but with sodium, it's hard to one, right? If I did exactly the same thing with sodium, then now all of a sudden, as the if I put Hodgkin-Huxley channels in, I'd see sodium concentration start to bump up with every action potential. That makes sense? All right. So we can do a lot in those 2 lines. And fundamentally, there's 3 types of questions that you want to be able to answer when you're doing these sort of reaction fusion. So where the dynamics occur, we've already seen that with the region. Who's involved, so what ion, sodium, potassium, chloride, magnesium if you want, I don't care, calcium. Proteins though too, right? Like calcium buffers. You know, I want to see what's the effect of this indicator dye. How much, if I insert a certain concentration of indicator dye, how much does that affect the wave speed? Well, and I can do experiments to try and extrapolate what happens if there was no calcium indicator dye. You know, and what is the reactions that's involved? So maybe there's a buffering reaction, maybe there's degradation. Certainly, phosphorylation is a common chemical reaction as well. All right. So that's the sort of the high-level overview. Just to break down into this a little bit, if I want to declare the basic units, basic usage for actually declaring a region, it's rxd.region, and you just pass in a list of whatever sections are involved. So this could be an actual honest-to-God list. You know, like we had a cell class, and so cell.all or soma.wholetree. This could be a manually enumerated list. Basically, anything that Python can loop over is cool. h.allsec, if you want to put it everywhere. If it needs to map to the intracellular space, in region I, you may want to define some cross-sectional shape. So if by default everything, we're going to assume that we're talking about the entire domain. But like I said, this is not what the cytosol looks like. Really, the cytosol kind of looks like this. There's all these holes in it where there's these organelles that are inside of the cytosol. So really, there's some fraction that it occupies. Another choice that people make from time to time is they're very interested in shells. So what's happening in that layer just below the plasma membrane versus the layer in the core of the dendrite? How are the concentrations different? There's actually this thing called the Frankenhauser-Hodgkin space. So that's sort of the area just outside of the plasma membrane. Again, it hasn't really equilibrated. There's no barriers per se, but it hasn't really equilibrated with the rest of the extracellular space. So locally, it's possible that the concentration is not the same as the concentration in the extracellular space writ large, just because it takes time. Diffusion takes time. All right, so that would be entering region O. And then, so then we maybe put a shell around everything. List comprehensions are fun. I have not uploaded these slides, but I will do this as soon as this lecture is available. As soon as this lecture is over, I'll upload the slides so you'll have all of these tips. But I don't want to get too hung up on any of this. So that's defining the region, defining the species. Generally, you're going to use RxD.species, and so that species, so it's chemical species, right? So like your calcium indicator dye, per protein, calcium itself, any of these things. And then what's the diffusion constant? Again, square microns per millisecond. I really should have included the units, but it doesn't. Those are the default units. If you want to specify an initial condition, you can just use comma initial equals whatever. So here, by the way, if initial condition is a constant, just put a constant. If initial condition depends on location, just put a function that depends on location. And I'll show you an example of how to actually do that and how we specify location in a bit. But you could imagine that the initial conditions is not necessarily uniform. Or another way to think about this, imagine you've done a simulation, you've saved your results, and you want to restore the simulation to some non-constant form, right? You ran a simulation, you saved it for a while, you ran a simulation, and then you want to just restore it from that point. You'll have a function that'll just look up the location and set the location in the right spot, right? And then that way you don't have to, whatever your nasty initialization is, you don't have to rerun over and over again. And yeah, you can give it a name. And as we saw yesterday, naming things makes your life easier, but it's not strictly necessary. One other little caveat here. So I talked yesterday about variable step integration. And let me just throw this out there. So the point of variable step integration is neuron chooses adaptively, continuously, the appropriate time step, right? So if the cell's not really doing anything, it can take a large time step because not a whole lot's happening. If the cell's in the middle of an action potential, it has to take a smaller time step because a lot of stuff is happening. And there's math that goes around that figures out the relationship, but really it ultimately comes down to the predicted error, numerical error in the integration. And here's the one little gotcha about this. Neuron's variable step solver has a default absolute tolerance of 10 to the negative 3. If I'm talking about voltage in millivolts, then I probably don't care about an absolute error of 10 to the negative 3 millivolts. Who cares about 1 microvolt? Doesn't matter. If, on the other hand, I'm talking about concentrations, all right, well, okay, neuron uses concentrations in millimolars. A typical sodium concentration, we said, was about 10 millimolar. So again, 10 to the negative 3, who cares if you're willing to allow that much of an error? If, on the other hand, I'm talking about calcium, remember calcium, we said, was much, much lower, at 100 nanomolars. Well, that's not going to work for me because an error of 10 to the negative 3 is 1 micromolar error, which is 10 times as much as calcium should be. So that's probably not so good. And so what Neuron allows you to do is it allows you to specify what's known as an ATOL scale. You can do this for your electrical states as well to help it figure out that a gating variable should have tighter tolerance than the membrane potential, for example. And if you're using the graphical interface, there are ways of doing this graphically, point and click with Neuron. But so from the programmatic side, here's how you do it with a chemical species. You just go ahead, when you define it, go ahead and specify an ATOL scale. So this says that maybe I don't want the default, but I want you to scale the default by 10 to the negative 6. And so now all of a sudden, now you do get you account for the fact that it's much, much smaller changes, and then that's going to give you a better stuff. Does that make sense on a conceptual basis? Things that are small, you need a scale factor, otherwise you'll have too much error. Only matters for variable step integration, not a problem for fixed step.
Is that something you have to specify for each species, or if you do it for one, it kind of applies and is consistent for all?
It's going to depend for every species, because every species concentration is potentially different. Like I said, sodium is, I don't know, what, 10,000 times higher concentration than, I don't want to use the same rule. Yeah. All right, so just as an example of initialization, so here if I want to define something, so here I create a cytosol. It's going to be everywhere that that's attached to the soma. Okay, don't need to do anything crazy with defining the cytosol. I have some species, so IP3, inositol triphosphate, is going to be present in the cytosol. I don't believe it's charged. I don't know why I put it charged, but who cares? It's not relevant for this example. And then there's an initial concentration. And so my initial concentration is not going to be uniform, but I want it to be a function. Well, okay, so I just define my function in advance, and then I just say initial is equal to my function. And so how did I do this function here? This function, I said, so define my initial of node. Node, you can think of, so you'll know, remember yesterday I kind of used node and segment interchangeably, and we've sort of talked about that. There's a subtle difference that occurs if I'm talking about three-dimensional simulations, wherein I may want a different discretization for the chemical stuff in 3D than I want for the electrical stuff, which doesn't have any real 3D variation. But for our purposes, node segments, they're all compartments. Okay. And if you don't like it, just ignore it, and you probably won't go wrong if you ignore it. But in any case, so I pass in the location, and then so now I just compute the distance. So right, what's the distance along the cell? So h dot distance. We didn't talk about this yesterday, but this is the path distance from point A to point B. So the path distance from the center of the soma to the node, and then I can return any function based on that if I wanted to. So basically, a hyperbolic tangent, as in case anybody doesn't remember their hyperbolic geometry, is basically a sigmoidal function. It's low, it's high, and it gets a little curvy in between. And so there you go. So now when things are near, things that are near the soma are near 0, or things that are far from the soma are near, I guess, 2. And then that divided by 1,000 sort of stretches the curvy bit, connecting the two. Does that make sense? So there's an example of, and there's, I believe this is available as one of the examples in the read the doc, so I also don't want to belabor that point. But I could also just grab the x coordinates. So somebody was asking me yesterday about 3D location. So it's not quite this convenient with an actual segment, but for if it's a node, I can just grab the x3D, and that's going to be the 3D coordinates of the point. And then so then I can make a statement where here this is sigmoidal based on the x coordinate location in 3 space. There's other things. So there's an RxD.parameter. If you wanted to declare something that varies with space, that you just want to store values in, they're constant, they don't change. You define them with an RxD.parameter. You can use these values in a chemical reaction as you want. They just don't get to, you know, if you have something combines with something, what doesn't go away because it's constant. If you want to plot stuff, you can use range bar plot. You just pass in these objects, the parameters or species or whatever, to a range bar plot, and you plot the concentrations along the path in that way. So parameters are like species, but they don't change. There's also a state variable. So if I have a gating variable or something that's not diffusing, right? Gating variables don't diffuse, but they do change over time. So I use an RxD.state for that. This is in the slides. We'll talk about it later if it comes up. Fundamentally, RxD.reaction is how we specify the reaction-diffusion concentrate, the reaction aspects of it. So if I have CA and buffer goes to CA buffer at a forward rate of KF and a backwards rate of KB, basically the syntax is the left-hand side, the right-hand side, the forward rate, the backwards rate. And you can always do it. Remember, Python has this built-in help functionality, so if you forget what the order is, you can say help parentheses RxD.reaction, and it'll tell you what's there. If you forget, wait, what was that thing called within RxD? Just do dir of RxD, and it'll tell you all the functions that are available. If you're using Colab, of course, it gives you those auto-completes, which makes life very easy. By default, it's assuming mass action kinetics, but you can specify mass action being false and then just give it the formula instead. You can also specify rates. So if you want to say IP3 decays at a specific rate, so reactions conserve mass, but if I'm talking about degradation, then I really don't care. I start out with IP3 and it degrades over time by some enzymatic reaction. I don't necessarily care about the debris. I don't need to keep track of the debris concentrations, right? Technically, it didn't just go into the ether, but if it's not going to go back, I can use a rate and say that, well, however much I have, there's some portion of it that's going to disappear at any given moment, right? And so then that would be a degradation type of reaction that you do with the rate. The other thing that you can do is a multi-compartment reaction, which is basically a pump. Right, so normally reactions only care about what happens in one location. Because if I have a molecule over here and a molecule way over there, they're not interacting because they're not together. They have to bump into each other to react. But sometimes you might want to think, well, I have a pump, it's binding to a thing here, it's binding to a thing here, and then it's doing a conformational change that flips. Well, then that is a different beast, and I do actually care about the concentration here and there. And so you can specify this with a multi-compartment reaction. But it's the same thing the syntax is the same as a reaction, except you just need to say what your regions are. So like imagine something moving from the ER to the cytosol. So a circuit pump, right? A circuit pump is going to, on the ER, is going to grab calcium in the cytosol and flip it inside and bring it into the ER. Conceptually, does that make sense? All right. You can get a list of nodes just like you can get a list of sections. So if you have your species is a protein, then protein.nodes is going to be the list of all your nodes, and then you can filter that using by passing in a region or a segment or a section or whatever to just give you all of the nodes that are in one specific location. So if I have calcium as a species, Ca parentheses ER is going to be all of the calcium nodes within the ER. What's within the calcium nodes that are in the ER at the center of the soma, right? Which is different from the calcium that's in the cytosol at the center of the soma. So there's more than one. So the spatial location is not just a function of position in the electrical sense. I can't just say the segment, but I need to say which, right? Because there's calcium in the ER and calcium in the cytosol. I don't know which one unless I explicitly say. Okay. We'll find out how much I've confused people later, I guess. But in any case, if I want to record it, so dot concentration would tell me the concentration. So dot underbar ref concentration gives me the concentration pointer, and then I can feed that into a vector record, same as for any other pointer. Right. At the end of the day, they're just pointers. You can grab surface area and volumes and all sorts of fun things like that. Pointers, just throw pointers into things. That's it. All right, so here's an example. Okay, so I've talked a bit, but we've covered all the things. We've covered the who's involved. We've covered the where things happen. We've covered who's involved. And we've covered what they do. That's it. Those are the 3 questions. So here's a full example, right? So if I'm looking at calcium buffering, and that big old star says, no, calcium buffering doesn't necessarily work this way. But let's just suppose that this is, so effectively this is the calcium chloride story, right? But let's suppose that I have a buffer that needs 2 calcium ions to bind at the same time. That's not really how things work. They're more likely to bind sequentially. Let's pretend it's simpler for now, right? But with that caveat, so I have a calcium buffer. I have a buffer that's going to bind with 2 calcium ions. They're going to give me the bound form, and the buffer is going to degrade over time. Well, I'm going to start out from neuron import H, but I'm also going to want to do RxD because I'm talking about the chemical stuff. So from neuron import H RxD, I'm going to want my units. You'll notice here I'm importing nanomolars instead of millimolars because calcium is at a very low concentration. I need to say where things happen. Well, I'm going to have a soma. Let's say I got to have a space, so it's an H dot section. I'm going to define the cytosol that lives inside the soma. Site is RxD region, wherever it is. So soma, known regions I. Cool. Who's involved? I've got calcium, which is a thing with charge 2, and has some initial concentration. I have a buffer, which has some initial concentration. I'm going to assume the buffer is uncharged. And I have the bound form, which has some initial concentration. And then I can say that, okay, well, I've said where things happen, I've said who's involved. Now I can say what's going to happen. Well, it's going to take 2 calciums, and a buffer is going to give me the bound form. And there's a certain forward rate and a certain backward rate. And the buffer, well, the buffer is just going to disappear over time. Actually, no, there's a typo here. What's the typo here? Anybody see the typo?
Should be a negative.
Should be a negative. Yeah, this is a buffer that increases over time. It was a negative when I ran it. This is just me typing. Okay. In any case, there's a buffer that disappears. And this is the full model specification. This is all the code that we need to simulate these dynamics, which are things that are happening presumably inside some neuron section. And then I could go and using all the stuff that we did yesterday, load std run.hoke, initialize my simulation, set some vectors up to, set some vectors up to record, then initialize my simulation. The vector recording has to be set up before you initialize the simulation, pro tip, or it's not going to work. And then plotting it. And then that's what I get. So this is the full model specification. Everything else is just make the pretty picture. It wasn't so bad. We did an awful lot. We defined our own dynamics. We didn't have to try and make any sense of mod files or anything like that. Everything is at a perfectly happy, friendly Python case. And I think you'll see in a few days that you can also do this within NetPyNE directly. And then it's still basically a Python. I just want to take, so that was a simple example. So here is, maybe. Here we go. So here's an example, if it loads. If it loads as fast as I want it to. Okay. So here's an example of, I'd mentioned circadian rhythms. It's actually how I got into neuroscience. The very first conference that I went to was Society for Research in Biological Rhythm. So it was a circadian rhythms conference. But so in any case, this is using the Leloup-Goldbeater model of circadian rhythms. So this is a, this was a paper that was published, right? This is a very real model. From neuron import HRXD, I'm going to load, let me make this a little bigger. I'm going to load my model. Here I use Bokeh instead of Plotly because I did not create this example for this course. But it's just a different graphics library. The same thing, right? I'm going to load my units from neuron.units import. And now it's circadian rhythm. So I'm not talking about millimolars per millisecond. I got protein concentrations that are nanomolars again. I'm worried about things over the time scale of hours, right? And obviously millivolts. So here's some magic functions, and these are basically just cheats. You can this example is linked from the Read the Docs. So you can go here and grab these and copy paste. But you'll see what these cheats do, but they're basically just ways of declaring a parameter in a species in a shorthand form. But you can copy paste them. They're great for that. But in any case, I'm going to define the morphology. I have a cell. It's got a diameter and a length. And inside my cell, I have some region. I didn't really want to bother with cytosol versus nucleus versus whatever. So I'm just going to, it's R. I don't care. But so then I can define my parameters. And so this is straight from the paper. It was very nice. They had a table of parameters. I didn't have to convert anything, and I didn't have to figure out, well, okay, what is this and what is this and millimolars per millisecond? No. Neuron provides units. This is why Neuron provides units. Actually, literally, this example is why Neuron provides units, because I didn't want to convert it. This example is what led to that. But in any case, so we can take the units and we can put them in. We can take the species doing this. And so all this is doing, this is using that shorthand function. But fundamentally what's happening is we're calling declare species, and we're just constructing a whole bunch of RxD.species objects. But it was a shorthand to make it so that I didn't have to copy paste RxD.species, blah, 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 RxD.species, blah, blah, blah. This is a for loop. Literally, it's a for loop. That makes sense? All right. In any case, so then I have a whole bunch of reactions. And these are just formulas that I took out of the paper. It was good fun. But you'll notice, so this is the mRNA for timeless. I don't know how familiar you are. There's a bunch of proteins that are involved. There's a period protein. In this model, there's a period protein and there's a timeless protein. And so mRNA gets created, and then that gets then that leads to protein production. That suppresses the mRNA. Blah blah blah. But fundamentally, what's happening here, you'll see that I have a certain rate. The rate of change of mRNA, the timeless mRNA, is proportional to this mess, which is a formula that's from the paper. All right, so this is just a differential equation. MT prime is equal to blah. Well, it's not. This is a term. MT prime is equal to a bunch of things, but it includes this term. Right? This is one thing that's changing the timeless mRNA. Which type of reaction is this? Where have we seen this form of an equation before? Is this mass action? Is this Michaelis-Menten? Is this Hill? Is this Leprechaun? This is Hill dynamics, because I have a concentration that's being raised to the nth power. All right, so blah, blah, blah. You have a whole bunch of reactions. We can specify them. The point is, this is a real model. It turns out that chemicals and proteins can interact in more than one way. Right, so I didn't want to leave you with the impression that things were always very simple. So this is timeless, that's free, versus timeless, that's dimerized, I think. I don't know. You'd have to look at the paper for what all the variables stood for. Documentation is good. But in any case, T0 is going to T1 at this rate. T1 is going back to T0 at that rate. So these are all concentrations of proteins that are in various states. Chemical reactions, blah, blah, blah. We set up recording. We're always going to want to record time if we're trying to do a time series. Yesterday, we did things that weren't time series. We also did space plots. But I want to record a bunch of concentrations, so I'm just going to get all of my things. The nodes is going to give me all the segments. There's only one compartment, because I didn't define, I didn't set inseg. So there's only one, so I'll just grab the pointer to the concentration. and I'll record that. And I'll record a whole bunch of them. I'll initialize for negative 65 millivolts everywhere. The actual membrane potential, this is strictly a chemical model, so membrane potential doesn't actually matter, but you know, it exists, so negative 65 is good. It's plausible. Why not? What I'm going to do here is I'm going to use a variable step integration. Why am I using a variable step integration? Because if I use the default, Neuron's default is 1/40th of a millisecond, fixed step. I don't want to simulate a day. I don't want to simulate 72 hours at 1/40th of a millisecond. I don't need it. It's not happening. There's, you know, my rate constants are measured in, look at this. This is a rate of 0.01 nanomolars per hour. I don't need to think about 1/40th of a millisecond. So I'm not even going to bother making a guess. I'll just say, Neuron, you figure it out. And that's what H.CVOD active true does. CVOD is an integration method. And then so I just tell it to go ahead and plot everything. And so the syntax for plotting looks different from in Bokeh than it did for Plotly, but this is literally just whatever graphics library you want. You know, I have a vector, MP, plot the stupid thing. And so there we are. By the way, time, since time is a vector, just to be clear, time is a vector. It's always recorded in milliseconds. But in order to get it to come out, in order to get that to turn out into hours instead, I divide it by hour, divided by the scale factor. It's going to turn it into hours instead of multiplying. Does that make sense? Because I'm going the other way. Okay. In any case, so then here we go. So these are the protein oscillations over a period of hours. And you see they go up and they go down and they go up and they go down. And it's just a property of that chemical system. Does that make sense? Now, if I have electrical channels that are sensitive, and so fundamentally, if you look at one of those gene regulatory network diagrams, you'll see that these proteins are tied into some of the ion channels. And there's interactions between these proteins and the ion channels, which is not surprising because one of the fun facts that this does is this leads to firing rates that go up and down at a rhythm of 24 hours. So that was a whirlwind tour. Like I said, it's on Read the Docs, but does the high-level overview make sense? We'll go through much slower in the actual example in the afternoon. All right, let's see what else we got. So with, that's a problem. I've talked about calcium waves and calcium-induced calcium release. I don't think I want to talk about it now, but I have a paper with Sam Namotan, myself, Bill Litton, that looks at calcium-induced calcium release and basically how the calcium concentrations in the cytosol and in the ER affect the membrane potentials and affect a working memory network is what we ultimately look at. So there's a series of 2 papers, one that's just looking at calcium waves within a cell. So here, time is on the x-axis, position is on the y-axis, and I'm seeing elevated calcium just kind of spreads. But if the cell had been firing a bunch before, that meant that the calcium in the cytosol built up, which meant that the ER was able to sequester it, which meant that the calcium in the ER built up. And that led to an effect wherein, with the interaction of ion channels, you can look at the details in the paper, but the point is it led to, as the concentrations changed, it changed how long a calcium wave, once triggered, was active, because the ER is now different, and it changed the electrical activity, and you see in the top diagram here the membrane potential. In the naive case, It stays hyperpolarized for maybe a millisecond or something. No, this is seconds, actually. It stays hyperpolarized for maybe a second, versus in the prime case, where the cell's been firing before, it stays hyperpolarized for a second and a half. And that may not sound like a lot, but now that's a form of non-synaptic plasticity. The cell is doing something different for a period of half of a second. If your cell is firing at 30 hertz, that's 15 action potentials that are affected by the hyperpolarization. You could imagine that the hyperpolarization means that the cell's less likely to want to fire, right? And how does this work? There's like calcium-activated potassium channels and things like that that allow these chemical concentrations to affect excitability. We actually looked at this in a separate paper. Again, I just refer you to the paper, and you'll have the slides of a working memory, well, persistent activity network. And the punchline is, it looked like if the ER calcium concentration was the dominant effect, we'd see more of a difference between the pattern activity that we want, the cells responsible for a pattern of activity that we want to maintain, versus cells for a pattern of activity that we are not trying to remember. So calcium had, the intracellular calcium dynamics had this stabilizing role on a working memory network, on a network of the type of working memory. I'm trying to avoid making any explicit biological claim here, but does that make sense? So there is a role for the interaction. The intracellular dynamics affect the stability of an individual cell, and that therefore affects the behavior of the entire network. Whether this matters to your network or not is another question. You have to decide if these dynamics are important for your story. They might not be. They might be. And half of modeling is deciding what not to include. All right, in the couple of minutes that we have left here, let me wrap up by pointing out that all of these dynamics don't just happen inside of the cell, they also happen outside of the cell, right? There are chemicals outside the cell. There are proteins outside of the cell. And so to define these regions, we use what's known as RxD.extracellular, and then we just define the region, you know, by spatial coordinates, X, Y, Z's. We use Neuron only supports a relatively coarse grid at the level where we can collapse all of the details about the blood vessels and everything, and the tortuosity and volume fraction. But once you have this is just a regular region. You use it the same way that you use any other region, and then you can you define your region, and you can, here's one way of just sort of plotting it. I'm plotting a slice overlaid on my cell, so I'm seeing an area. What was this? I think this was an example where I was just having the cell release some material at a constant rate across its surface area. And so then obviously where there's more surface area, we're going to see higher concentrations. It's going to give us, it's what's giving us the cell as a glow. Let's not worry about this too much, but here's the context of why this matters. So what we did, this was a fun one. We took a bunch of cells. They're not strictly Hodgkin-Huxley cells. There's more of a persistent sodium has a higher rate of activity here. But in any case, we took a bunch of cells of Hodgkin-Huxley-esque type. And then I said, well, suppose for a moment that the extracellular potassium concentration was elevated in one specific location. And then I wanted to see what would happen. And so these X and Y coordinates are, so this is a slice of the brain. Each dot is a neuron that's been distributed randomly. And they have sodium and potassium channels. So what happens if the extracellular potassium concentration is elevated? Just in general, what happens to the extracellular potassium concentration is elevated? Let's put it this way. Suppose that the extracellular potassium concentration is elevated to the point that it is the same as the intracellular potassium concentration. Then what happens?
cells would find it hard to recover. from
Right.
that.
Membrane potential would go up, but potassium couldn't bring it down because there's no net difference. So there's no movement across the membrane. So potassium wouldn't be able to bring it down. And if we're not quite that extreme, as you say, maybe they are able to bring it down, but it's going to be harder. Does that make sense? So as the extracellular potassium concentration goes up, the difference between the outside and the inside goes down, which is to say that it's going to be hard for potassium to do its thing. So now here's a question. Suppose that I'm, below the point where they're equal. They're not equal, but that it's higher than normal. So the extracellular potassium the potassium channel is going to have a hard time bringing the cell's membrane potential down. Which is to say the potassium channel is going to have to stay open longer, probably, right? The cell is going to be depolarized longer because it's going to have a hard time pulling it down. Potassium channel stays open longer. Then what happens?
Refractory period is longer.
Probably. I don't want to think about it. I'm not in any way disagreeing, but that wasn't. But in particular,
the potassium will go outwards for a longer time, so it will be self-sustained, kind of.
It'll be self-sustained. So we ran this, and let's see what happened. So we started with the region of elevated potassium, and we just ran this, and we saw what happened. And so it just spreads. We do have some mechanisms, I think, inside that are clearing it. But in any case, we got a wave of spreading extracellular potassium concentration. And if you're wondering why it's not perfectly circular, it's because the cells are positioned randomly. They're actual cells in actual locations because this is neuron. We're not just sort of blurring everything in a mathematical continuum model. All right, but we get this regenerative wave. So now we've talked about waves that exist electrically. We've talked about chemical waves within cells. We can even have a chemical wave outside of cells. And as was mentioned, this is going to affect their electrical firing. I didn't want to think about that earlier, but it's going to affect their electrical activity as well. Okay. And finally, as a final point, so neuron does that 3D extracellular. You just define the region, and everything's fine. In neuron, if you want to do everything in 3D, you can do the inside in 3D too. Why not? And all you do is you have this one extra line that you add at the very beginning. RxD.setSolve type dimension is 3. If you give it a list of sections, I care about this part of my cell in 3D. I don't want to do the whole cell in 3D. I care about this part of the cell in 3D. And it might matter if you're talking about where the dendrites meet the soma. It might matter if you care very much about spines and you're focusing on spines and dendrites. And spines are very small relative to dendrites. And you want to see how things are going. Maybe you want to do those in 3D. You can also do it in parallel. And so what happens is we take everything, we sort of discretize it down, we get, we turn that geometry into a bunch of boxes. And then so for each box, we get one concentration. So we actually have multiple, this is why it matters that nodes and segments aren't quite the same thing, right? So for one, you might have, this whole thing might be one electrical segment, but the concentration here is going to be different from the concentration there. Because chemicals, well, goes like this, right? Chemical diffusion is much slower than electrical signaling. So we care about this in space, and this sort of matters. So I've got a wave coming in. So each one of these is evenly spaced at the same time point. I have a wave coming in here of elevated calcium. All the dynamics everywhere are exactly the same, but now you get to a case where you have a small source feeding into a large source. Electrically, this could lead to problems with impedance. Chemically, what's going to happen is it's going to be hard if it's a regenerative phenomenon, we have to raise the concentration high enough, but there's now so much more that things could diffuse away than build up. It's really hard for it to enter into the big space. So it's kind of a chemical story the chemical analog to the electrical story of impedance mismatch. that could block a signaling. Here, this one doesn't block the signaling, but it does stall it, basically at the entry to the soma in this particular experiment by 65 milliseconds, which again, in the course of action potentials, is nothing. Oh, I'm sorry, in the course of action potentials, that's a long time. Here's a full example. It's in the slides. I'll upload them shortly, but this gives us, you know, this is literally, here's code. It runs a 3D simulation. It fits on 2 pages, 2 slides. And you'll have this code. And it, more importantly, not only does it run it, like half of the code here is, and make the pretty picture. So contour maps are a great way of doing this. Another way that I like to do this is to use sort of level sets and do, so Plotly does these level sets, these 3D level sets. And so if you make them translucent, then you can sort of see inside of objects and see the curvature, the wavefronts inside of objects. So I have, I think this is basically the same simulation, but with that kind of a visualization. I've already mentioned you can do slices. Here's another view of, like, I've got a 3D point source. So if I'm doing a 3D simulation, I care maybe not just that the input is coming into this particular segment. I may care very much about, well, is it here or here? It doesn't matter that much electrically if it's just an ionotropic receptor, but if it's a metrabotropic receptor and I've got something here and something right next to it that are going to interact, that's different than something on the opposite side of the dendrite. So we can do those sort of simulations in Neuron. You need to use a mod file to receive the event, and then you can just pick wherever you want to put it. So that event, that synaptic event, leads to the production of a material here, and then it's going to diffuse away and then stabilize. And then so we're seeing different time points as the thing spreads out and diffuses. And we'll look at examples, well, not this complex, but we'll look at some examples of this in the afternoon. We've got a few articles on this. Again, all these links are in the slides. And then we'll do 3 examples. They're all set up as Google Colab notebooks already, I think. So they'll step you through problems. And we'll do those in the afternoon. All right. And with that, I thank you for your attention. I'll hang out if anybody has any extra questions for a bit. And I think we come back at 2?
At 2, but maybe let's do like an hour, so come back at 2:10.
Okay, 2:10. All right, thanks everybody.
Thanks.
Thank you, Robert.
Thank you very much.